Okay, so for genetic engineering, you actually don't have it in your supplementary books. So I'm going to be sending you the material for this topic. But it's a very fascinating topic because it has so many applications, especially to what is happening now. The fact that we are all at home, um, and we believe that it's due to a new virus that possibly could have been genetically engineered. So we really want to explore what it's all about, how do we do it, what are the benefits of doing genetic engineering, what are the dangers associated with it, what are the cons, what are things that we have to be cautious about. And as I said, it's really about, this topic is mostly about discussion, and the content about the content, I'll make it available to you. So all the documents that you will see today, I will send them to you so that you'll have them. So the first thing you have on the screen is that it's a video introducing you to the concept of what genetic engineering is all about. Now, we have done natural selection and we have done artificial selection. And when we were doing artificial selection, we basically said that we could design animals or have animals that had the traits that we wanted, that we found to be beneficial to us. But when we were doing artificial selection, we were only able to do it by mating the organisms with each other. So if you needed, say for example, you wanted to create a breed of cows that had superior milk, then you would have to make sure that you found a female cow that had good milk with you'd mate that cow with a bull that you thought had the traits you wanted, and then you would watch the offspring that they produced, and you would observe the offspring to see which of the offspring actually has the superior milk quality that you want, and then you would allow that offspring to breed again, so that they can produce another set of offspring that has the superior qualities that you want. But basically with artificial selection, we had to keep breeding the organisms that had the traits that we wanted, and to produce a pure line, or a pure population. But genetic engineering has basically changed all of that, because genetic engineering allows us to identify a trait that we might, say for instance, we might find a trait present in the horse. And if we think that that trait would be beneficial to be present in our cow, we can actually do this by genetic engineering. So, and the horse and the cow are of two different species, so we couldn't get them to mate with each other. But with genetic engineering, we can bypass that entire process of having to mate them with each other. And even though they are of two different species, we can cut out the gene that is of interest to us, and we can insert it into the genome of the next species that we want it to be present in. So with genetic engineering, we are able to surpass the boundaries that we had before with artificial selection, and we can mix the traits from one species with another species, and it has opened up endless possibilities to us, and it also has created unforeseen problems as well. So the first thing we're going to do, I'm going to show you this video, and this is just to give you an introduction as to what this whole process is about. You don't necessarily need to write anything from it, because as I said, all the notes that you need, I have already have them compiled in documents, and I will send them to you after the class. And I would need for you also to reread this information that I will send to you, so that when we have our next class, we can actually go into doing questions as it pertains to it. So watch the video first. It's an introduction, and then we'll get more, we'll get deeper into what genetic engineering is about, okay?
Hi guys, in this video we'll be looking at an introduction to genetic engineering, the overall process, and then we'll finish with a summary. So with our advancement in DNA techniques, we've begun to discover ways of genetically modifying organisms. A genetically modified organism is sometimes written as GMO, and this can refer to both animals and plants. So for example, some animals that have been genetically modified include mice or rats, and we've got several examples of genetically modified plants that we use as well. So a genetically modified organism is an organism which has had its genome altered by genetic engineering techniques. So remember, the genome refers to all of the DNA contained in that organism. And if we alter the genome ourselves using genetic engineering, then we've genetically modified that organism. Scientists develop GMOs because these organisms tend to have a more desirable phenotype or several phenotypes. For example, we've genetically modified some bacteria, so we've altered their genome so that they have a desirable trait that produces something that we use. For example, they can make certain drugs or hormones that we can use in medicine, whereas the unmodified bacterium wouldn't do this. Another example is a genetically modified plant, which is the strawberry plant, and we've genetically modified it to sometimes become drug resistant, which means that when we spray pesticides and insecticides over particular farm areas, the actual strawberries are okay and they won't suffer from the drug. The genetically modified organisms can be made by taking a gene from one organism and inserting it into the genome of another organism. So for example, organism 1 may have a gene which is desirable for a particular product. So for example, this might come from humans or a particular animal. And then what we do is we insert this gene into organism 2, which is often a different species, for example, a bacterium. And you can see we can insert the gene into the bacteria's genome, and then this bacteria has now become genetically modified. When we carry out this process, this specific type of GMO is called a transgenic organism. So transgenic because it's referring to genes, and trans tends to mean transform or changing. So we've taken the gene from another organism and we've created a transgenic organism. A DNA molecule which contains DNA from more than one organism is then called recombinant DNA. So if we took the gene and we inserted the gene from one organism, number 1, into organism number 2, then this DNA is now called recombinant DNA because it's almost been recombined to contain new DNA from another organism. So what this means is that a transgenic organism is an organism which contains recombinant DNA. So it combines the two terms. Transgenic meaning that it's taken genes from another organism, which means its DNA contains not only its own genes, but also DNA from another organism. So it's recombinant DNA. Transgenic organisms can successfully express a gene from a different organism because the genetic code is universal. So the genetic code basically refers to how DNA and its nucleotides codes for a protein, because certain nucleotides in groups of codons code for the same amino acid. This is universal, which means that it's basically found in all organisms that we can study: prokaryotes, eukaryotes, protoctista, and all the different kingdoms that we've seen. So because of this, what this means is if we take one gene and place it from one organism into another organism, because the genetic code and the way the code is read is the same, that organism can read that code just as it would its own code and make the protein. And in doing so, it expresses the gene. So this is a really important, useful trait that we can use to modify organisms and allow them to read code from other organisms. So we're going to go through the overall process of genetic modification of an organism. The transgenic organisms are created using the following stages. So first of all, we get the gene of interest, the gene that we wish to be transferred to another organism, and we have to isolate it so it's on its own from the original organism. So for example, this is a cell of a particular organism, say organism 1, and there's a particular gene in that cell which is desirable. We isolate that gene so that we've got it completely on its own from end to end. So this gene makes a desirable product, for example, a drug molecule or a hormone. Now what we need to do is think about how we get the gene into the second organism. The gene has to be placed inside something called a vector, and the vector carries the gene. So essentially here we have our gene, and this is our vector over here. Essentially we just insert the gene into the vector. So what we're left with is the vector now containing the gene and holding it within its own genetic material. So what is a vector? A vector is something that's capable of carrying and inserting a gene into the host organism. They often are plasmids, viruses, or bacteria. So remember, a plasmid is a ring of DNA found in bacteria. So the diagram just above used the example of a plasmid. So this is all DNA, and all we did was insert the gene into 2 points of that plasmid to make it larger and make it contain that DNA. Viruses obviously are types of pathogens, as are bacteria, so they can all be vectors. So by this point, the vector contains the gene, but it hasn't managed to be transferred to the second organism. So next, the vector inserts the gene into the host cell. So we took that plasmid out, and we inserted the gene into the plasmid, and now we have to insert the plasmid into organism number 2. So at this point, the bacteria takes up the plasmid, and because it's taken up this recombinant plasmid, it's got this extra gene that we put into the plasmid too. The host cells which have successfully taken up the vector need to be identified. So in theory, all we need to do is put the vector into the bacterium, but this isn't always easy. Sometimes the bacteria don't take up the vector, just because of chance or bad technique or errors. So sometimes the bacterium just doesn't take up that vector. But then in some cases, we're successful. It does take up the vector. So what we need to do is identify those that have taken up the vector against those that haven't. And then the only ones that are useful to us are the cells which have taken up the vector. So the cells that have taken up the vector express the gene of interest. We don't grow them or clone them. So now we've identified those ones that have the vector inside of them, and so they've taken up that gene. So now we just need to multiply the cells in either a culture or some sort of cloning technique so that we have lots of copies of this organism that's been genetically modified with that new gene. Each of the stages that we've mentioned can be achieved by using a variety of different methods, and there are lots of different processes for different examples of genetic modification. Hey guys, I hope you enjoyed the video.
Alrighty, we're going to go back through this step by step now to dissect what exactly is being said, because I want for you, when we're finished, I want you to have a mental idea of exactly all the steps you would need to do if you wanted to genetically modify an organism. All right, so let us go back through what was said step by step. So the first thing was said to you was, what exactly is this process called genetic engineering? And it says here that it's an advancement in DNA techniques that have allowed scientists to develop genetically modified organisms. Next, now it explains to you, what does it mean really when we say that an organism has been genetically modified? So it says that genetically modified organisms are organisms that have had their genomes altered by some form of genetic engineering technique, meaning that you remember that the genome speaks to all the genes that you possess in your cell. So if you have added a gene to somebody's genome, then you have altered it. And that would have been done by genetic engineering in the lab. Now, why do we even need or even explore this? It's because we believe, or the scientists believe, that by doing this process of genetic engineering, you could actually introduce desirable traits into organisms, right? Making them better than what they are. No. So for example, as you're seeing on the screen, like the strawberries and so, you could make them become sweeter. You could make them have a longer shelf life. And for example, bacteria, you could modify them so you could get the bacteria to produce drugs for you. You could get the bacteria to produce insulin, antibiotics, and various substances. So they believe that this technique would have been beneficial to us because we would have been able to insert beneficial traits into the organisms. Okay. So once an organism has had its genome altered, then we say it has been genetically modified. So genetically modified organisms is basically produced by taking a gene. And remember that a gene is a portion of DNA that codes for one polypeptide. So if you take a gene that can code for one polypeptide from one organism and then you insert that gene into the genome of a different organism, then that is what we call, well, that's what we consider genetic engineering to be. And usually you're taking the gene from one species and inserting it into another species. As you are seeing here, so this would be the gene from another species, and you're inserting it into the bacterium right here. Okay, so once you have done that, then you can say that you are doing genetic engineering. Now, whenever an organism has been modified by genetic engineering, it is now known as a transgenic organism. So once it has a portion of its DNA coming from a different species, then we say it is transgenic. Now, another term that was introduced to you is that whenever you have DNA that has been created from more than one organisms, then we call that DNA recombinant DNA. So in this case here, you can see the bacterial DNA is in blue, and the gene, the one that is in pink was this new gene that was inserted. So now, because this piece of DNA has two different sources, it is comprised of DNA from two different sources, then we consider it to be recombinant DNA. So those are just some terms that you have to be aware of. Okay. And then when we said that a transgenic organism would have been one that was genetically modified. And another thing being pointed out to you here is that a transgenic organism is an organism that does contain recombinant DNA. So the organism has DNA that has been modified to include DNA from another source. Okay. Now, transgenic organisms, now, the reason why this whole thing of genetic engineering can even work. So the only reason why genetic engineering is even possible or even plausible is because the genetic code is universal. So when we were learning about DNA, and we were learning about structure, and we were learning about the genetic code, one of the features of the genetic code was that it is universal. It is one language spoken by all the different species on the entire planet. So the same sequences that are used in bacteria can be read by the trees, and they can be read by the fishes, the humans. So all the different organisms on the planet, we use the same genetic code. So I can take a gene from a virus and I can put it into the bacteria, and the bacteria can read the information that is present in the gene. And if it was a case where the genetic code was not universal, then that would not have been possible. So after they discovered that it is universal, that is when they realized that genetic engineering was possible. You could take any gene from any organism in any species, and you could insert it into any other species that you desire to do so with. Okay. And next thing now was what exactly was the overall process of how it is done. Now, this is the interesting part, and I want you to pay close attention to this, and I'll go through it another time. Now, when you want to genetically modify an organism, then there are some steps. The first thing you must do is to find the gene that you want to insert into the next organism. So for example, say you wanted some bacteria to start making insulin for you. The first thing you would need to do is to find which chromosome in the cell contains the insulin gene. Remember that we have 46 chromosomes arranged in 23 pairs. You would have to learn which of those 23 chromosomes actually does contain the insulin gene. Not only that, remember that a chromosome contains multiple genes on it. So you would have to search that chromosome now and find out where exactly on the chromosome is your insulin gene. And then you would have to be able to isolate it. So meaning you would have to be able to use enzymes to cut out that section that you are interested in from the chromosome that you are interested in. So that is the first step of the entire process, which is to find the gene of interest, isolate it from your original organism, as you're seeing there as your step one. So now, normally to do this, you must use specific enzymes. So he didn't mention the name of the enzymes, but in the next document that we will look at, you will see the names, because the names are important for you to remember. Now, the enzymes that will be able to cut out your DNA or your gene for you, they are known as restriction enzymes or endonucleases. These enzymes have the ability to cut the DNA, and they cut the DNA in a zigzag pattern like you're seeing here. So there's this part of the DNA that is single-stranded, and also over here is single-stranded. So we say that it is in a zigzag pattern. Now, the way that the enzyme cuts the DNA and leaves it with these two ends that are single-stranded, it actually is advantageous, and it makes the DNA have sticky ends, meaning that it's easy for you to now take this DNA and insert it into another DNA because it has these sticky ends that are left out, right? So enzymes are the ones that cut out our gene of interest for us. Then next, the next process now that we need to find something known as a vector. So we need to put that gene of interest into another structure that has DNA that is known as a vector. Now, the vector basically is going to take our gene up and it's going to take it into the organism that we want to put the gene into. So it's just like a vehicle to get our gene into our organism of interest. So a lot of times, one of the commonest, let me see, somebody has something in the chat here. Nope. So Olivia's asked if I'm given notes. No, we haven't given notes yet. So one of the commonest vectors that we use in order to insert our genes into so that they can take it into the organism of choice is we use these circular pieces of DNA called plasmids. Now, plasmids, these circular DNA that you're seeing, are usually taken from bacteria. So bacteria have two sets of DNA inside of them. So the bacteria will have their regular DNA, and they also sometimes have an additional set of DNA in a circular format known as a plasmid, like what you are seeing here. Now, these plasmids are excellent vectors because we can take them out of the bacteria, we can cut them open, and we can put our gene of interest into them. And then once we have done that, we have created a recombinant DNA. So when we put this DNA inside of the plasmid now has DNA from two sources, and it is now called recombinant DNA. So now once we have a vector that has taken up our gene of interest, then the next thing we need to do is to get that vector into our cell of interest now. Now, we don't only use plasmids as vectors. We do have other types of vectors that we can use as well, which he mentioned. So it says that the vector is something that is capable of carrying and inserting a gene into a host organism. Vectors are often plasmids, viruses, or bacteria. So plasmids are the most common ones. Viruses can be used as well because viruses can infect bacteria or other bacteria, but plasmids are most often used. Now, after we have gotten our gene of interest into the plasmid, the next thing we will need to do is to get that vector into our organism, right? So the vector is then inserts its gene into the host cell. So usually our host cell is a bacterium, right? So this is a plasmid. Remember I told you before that the bacteria has two sources of DNA. So this is the regular circular DNA that the bacteria would have, and this is the plasmid. But this plasmid has been modified by us because we have inserted a gene into it, and we have gotten the plasmid to be inserted back into the host cell. So once we have gotten our recombinant DNA into our host cell, we would have said that this cell is now transformed. So whenever you have a bacterium or a cell that now has taken up recombinant DNA, it is now considered to be transformed. All right. So let us continue. So this was the bacteria before it took up the plasmid, and this was after it took it up. So after it takes it up, we say it has been transformed. Now, not all of the bacteria are actually going to be able to take up your recombinant DNA that you're trying to give it. So you know, you also have to find a way to figure out how much bacteria has actually taken up your gene. All right. So sometimes you don't get all the bacteria to take up your plasmid. You have to, some of them will take it up, some will not. And then you have to also check to see which bacteria took it up. So there is this process where you try to see which bacteria have been transformed, which ones have taken up your plasmid. Now, to check to see which bacteria have been transformed, there are various ways we can do it. Sometimes what you do is that apart from just the gene that you want to get inside of your plasmid, sometimes you can add another gene to it that can help you to identify your plasmid if it has been successfully taken up by the bacteria. So a lot of persons, what they will do is that they will put a gene beside your gene of interest, like antibiotic resistance, right? So what they will do is that they will check to see if the bacteria has taken up the plasmid by treating the bacteria with a specific antibiotic. Now, all the bacteria that had taken up the plasmid would have the gene for antibiotic resistance. So if you treated them with the antibiotic, the ones that have taken up the plasmids will not die, they will survive. And the ones that did not take up the plasmid will be susceptible to the antibiotic and they will die. So a lot of times, you have to place some other gene along with the gene that you're interested in so that you can use it to check to see which of your bacterial cells have actually taken up your plasmid successfully. So you normally have to do that process where you check, right? Now, once you have identified the ones that have taken up your gene of interest, then you will allow these to multiply rapidly. Bacteria multiply rapidly. They double their population literally every 30 minutes. So you can start off with one bacteria and in a day you'll end up with millions of bacteria. And remember that bacteria they reproduce by mitosis. So the daughter cells are going to be exact copies of the parent. So once you have identified the ones that have taken up your gene, then all the offspring will be recombinant. They will have the same recombinant plasmids and they will be able to produce your gene of interest. So after you've allowed your bacteria to be cloned, then at this point, you just wait and let the bacteria actually read the gene and then produce the protein that you're interested in. And then normally at this point, once the bacteria have produced the protein that you're interested in, then what you can do at that point is kill the bacteria, purify the protein, and then you would have collected the protein that you are interested in. All right, I'm going to ask you to give me 1 minute. Let me see if I can help out Gabrielle because she doesn't seem to be able to get inside the group. So let me just send her an invitation. Back again. So this is an overview of essentially what the entire process of genetic engineering is about. But as I said, there were some little specific details that, he didn't go much into that I want you to know the actual details of. So I'm going to bring up that for you now. And as I explained to you earlier, all the documents that are going to be shown today will be sent to you, okay? So I really do want you to focus on grasping the concepts of what we're doing because all the information will be sent to you. And it's not about the writing notes per se, it's about understanding it and being able to use information appropriately, okay? So I'm going to go through this document that goes through the process another time, similar to what we just did. And when we're finished, I'm going to just ask to see what you have gathered from the entire thing. Just checking. Are you able to see the document on the screen?
Yes, miss.
Okay, great. So genetic engineering, all right, as we had said from before, is when you insert, as it says here, genetically modified organisms are organisms that have had their DNA altered through recombinant DNA technology. And recombinant DNA technology involves the transfer of fragments of DNA from one organism or species to another. Let me highlight that, okay? Now, transgenic organisms can successfully express a gene from any organism, as the genetic code and the mechanism of protein production are universal. So again, we talk about the process. So the first step of the process, which we had said from before, was that we need to create the fragment. We need to isolate the DNA that we're interested in. So how do we do that? So we would use restriction endonucleases. So these were the enzymes I was telling you about that you needed to remember the names of. So we use restriction endonucleases to cut at recognition sites near the desired gene. Or if that can't work, all right, we can do another process. So the scientists have become very smart. Now, if you can't actually cut out the gene that you want, what they have discovered is that if you found the mRNA of the gene that you desire, then you can create the complementary DNA from that. So this represents complementary DNA. Let me write it out so that you know. So they're saying that if you can't get through to cut out the gene that you want, and all you have is the mRNA of the gene of interest, that can work as well. What is happening to this thing here? Alrighty. So if you get the mRNA, you can convert the mRNA back into DNA if you use a different enzyme now. This enzyme is known as a reverse transcriptase enzyme. And then you can create your DNA from that, right? And another option too is that you could actually synthesize your gene by using a genetic machine. So if you actually know the protein structure that you're trying to make, you can work your way back and figure out what the DNA must have looked like, because you know the code for each protein, you know the code for each amino acid. So you can work your way back and say, okay, if I see this amino acid, then this must have been the DNA code, and work your way back. So there are several ways they are saying that you can get that piece of DNA that you want, all right? So various methods are there that we can use. But after we have done that, the next thing we need to do is isolate our gene, right? And normally we add we add some extra bases to our gene, which are known as promoters and terminators, right? Then, after we have done that, we need to get a vector. A vector is used to transfer the isolated gene into the host cell. And as I had indicated to you, that the main vector that we use all the time, most of the time, are plasmids, right? Now, in order to cut our plasmids open, we come back and we use the same enzymes that we use at the top. So the same restriction endonucleases that we use at the top when we were cutting our source DNA is the same one that we use to cut our plasmids open, and it creates sticky ends. So you must use the same enzyme that you use from the top to make sure that you use the same plasmid down the bottom. The same enzyme to cut your plasmid, right? So restriction endonucleases are used to cut the plasmids open, creating sticky ends. The same endonucleases isolates the gene, so the sticky ends of the desired genes and the plasmids are complementary. And then another enzyme comes in called DNA ligase that joins the two of them together. So we will have cut the plasmid with the same enzyme we use to cut out our gene, so that the two of them can seal together nicely. And then to join them up properly together, we use this enzyme called DNA ligase. So after we have done that now, we need to get at this point we can call it recombinant DNA, because once we have created a combination of, once we have put two pieces of DNA together, it's now called recombinant DNA. So we need to get that recombinant DNA now into our bacterial cell. So to reintroduce the DNA into the bacterial cells, the recombinant plasmid must pass through the cell surface membrane of the bacterium, and that is known as transformation. To get the plasmid to get inside of the bacteria, which is the transformation process that they just described, there can be several techniques that you can use. So you could mix the bacteria and the plasmid together in a medium that contains calcium ions, because they have found that calcium ions increases the permeability. And you can change the temperature to also make the bacterial cell surface more permeable, right? And once we have been successful in getting the plasmid to enter into the bacterium, then we can say that our cell has been transformed, okay? So once it gets inside, our bacteria is now considered to be transformed because it has taken up our recombinant DNA. And what we do is that we allow this bacteria to be multiplied multiple times so we can get many copies of our DNA. So this now, this part of it is what I was telling you about, that sometimes you need to figure out which of the bacteria have actually taken up your gene, because it's not all the time that they will take up the gene, right? So we have to check. All right. So transformed bacteria can be detected by using marker genes. For example, you can put two genes into your plasmid. So you could put a gene, a marker gene that is commonly used is a gene that is used to identify which bacteria have successfully taken up the plasmid. Now, it is an antibiotic resistance gene. That is a common one that we normally use. So you put a gene inside there for antibiotic resistance. So the transformed bacteria, they are identified because they can grow on a medium that contains antibiotic. The second marker that you can use to distinguish between bacteria that have taken up or empty bacteria would be when a recombinant plasmid is formed, you make sure that you put your desired gene in the middle of the second marker gene, making it non-functional. Therefore, the bacterial cells that express the second marker gene do not contain a recombinant plasmid. All right, I'm going to actually delete this because I don't want to complicate your life. So just learn about the antibiotic resistance, all right? So that's the common one that's used, antibiotic resistance. All right, so the other parts of this process now is that after we have finished making sure that we check which bacteria is transformed, then we make the bacteria multiply and produce our gene product by transcription and translation. And then the bacteria are killed and the gene product or protein is purified and collected. And then I'm going to go through two diagrams which illustrates this process. So the first one is a simplified version of it. So this one says, for example, we wanted to make insulin. We'd have to search and find where inside of our cells we have the human insulin gene, find the chromosome, and we would cut out that part of the chromosome, the gene, with a restriction enzyme. We would also get our bacterial plasmid, which is our vector, and we'd cut the plasmid with the same restriction enzyme that we used to cut this out with. Then the two of them will stick together naturally, but we seal them up together with this enzyme, which is ligase. Now this is our recombinant plasmid, and we get it to go through the cell membrane of the bacteria by the methods we saw earlier. We could use calcium ions to help it to get inside, or we could increase the temperature to help the plasmid to get inside. But once the plasmid gets inside, we say the bacteria has been transformed. But normally we need to check to see which bacteria were transformed.
Excuse
Yes?
me, Miss. I have a question.
Yes.
How does the restriction enzyme know exactly what it's cutting out? Is it that it's directed some way?
Very good question.
How does it know exactly what it's cutting out?
All right, so Desha, inside the body we have thousands of restriction enzymes, and the restriction enzymes actually identify certain DNA sequences. So it depends on what sequence it sees. So for example, some of them might see, once they see GGA, they will cut. That's what they're programmed to do if they see that sequence. So basically you will have to know the sequence of the gene, and then you would select from the pool of restriction enzymes the one that will cut that specific sequence for you. So that's how it works.
Okay, Miss. Right. Thank you.
No problem. So it's basically about, as I said before, you would know the sequence of your gene and you would select the restriction enzyme that can recognize that sequence. All right, so remember that we have to make sure that we check to see if the bacteria has actually taken up our plasmid. And usually we do this by making sure that we also inserted a gene for antibiotic resistance inside of the plasmid. So we will grow the bacteria on an antibiotic medium, and only those that took up the plasmid will survive, and they will grow and multiply. And then they will start making, they will start reading the gene, transcribing it, translating it, and create a protein that you want, and you get a purified sample of your protein. So that was one diagram. This is another simplified version. No, I'm a little bit more complex than the one we saw before, but I always like to give you diagrams of the processes so it helps you to explain yourselves better. All right. So another diagram talking about how you do the process is that you collect the human DNA, right? And you also get your plasmids from the bacteria. So you have to be doing step one and step two at the same time. You would use enzymes to cut the DNA of interest and the same enzyme to cut the plasmid. Then you would allow your gene and your plasmid to be mixed together, and they will stick together because once you cut them with the same enzyme, they're going to have the same sticky ends, or they're going to have sticky ends which are complementary to each other. So when they see each other, they are naturally going to want to stick to each other. And then remember that to seal it up properly, we use the ligase. So once we have now put our gene into the plasmid, it's now considered recombinant plasmid. We get the plasmid into the DNA, into the bacteria. That is transformation. Then we check to see which ones were transformed by growing them on a plate that has antibiotic in it, that only the ones that have the antibiotic resistant gene can survive. And then they will survive and reproduce and produce large amounts of insulin for us. Okay? So that is the process of genetic engineering, the process of it. And several questions, love to check to see if you can identify this process. So really, I'm sending you this document with the diagram so you'd be able to reproduce it, okay? All right, so I want to show you another video now to say, why are we doing this again? What is the benefit of us doing this genetic engineering? What are the good things that we get from it? What are the harmful things? And then I'll just give you a short break before we continue. So let me just pull up that one. So this one is going to talk about genetically modified organisms and what are the benefits and what are the harms. So I know you're not seeing it yet, so let me bring it up for you.
Genetically modified organisms, or GMOs, have had their DNA artificially modified by humans using modern genetic manipulation techniques. By changing the DNA, so changing the genome of an organism, means we can change its characteristics. Humans have been artificially altering the genomes of plants and animals for thousands of years by selective breeding. Today, with advances in genetic engineering techniques, means we can speed this process up by incorporating specific new genes from one species into a completely unrelated species. Crops, farm animals, and soil bacteria are three of the most common uses of genetic engineering to make GMOs. The cotton plant that grows the cotton for your t-shirt might have been genetically modified to be pest resistant, or the bacteria modified to produce life-saving medicines, as well as the better known GM crops that we eat. And then there's some wacky organisms, like spider goats and glow-in-the-dark mice. Firstly, let's find out how genetic modification happens. The short answer is, there are a couple of different ways. There are several techniques that can be used to modify a genome, but the main one relies on bacteria. Bacteria are single-celled organisms that don't have a nucleus like our cells, but instead have one main circle of DNA called the circular chromosome and lots of tiny circles of DNA called plasmids. In GMOs, it's all about the plasmids. They are really useful tiny bits of DNA that can contain one or several genes. The cool thing about plasmids is that they can be passed between bacteria as easily as shaking hands. So plasmids are a super important part of how genetic modification happens. Let's go through the steps using the human insulin gene as an example. First, we cut the gene for human insulin from a human cell using an enzyme. We then use the same enzyme to cut the circular bacterial plasmid open and stick the insulin gene and the plasmid together. Then we put the modified plasmid back into a bacterial cell, and we wait. But not for too long, as bacteria cells divide roughly once every 30 minutes. And a little while later, we have millions of bacteria producing the human insulin protein and excreting it.
The human insulin is filtered out and purified and can then be used by people with diabetes. How simple is that for saving lives? Spider goats are also a great example of a genetically modified organism. They aren't as scary as they sound. They're goats that have had the spider silk gene inserted into their DNA, so when female goats lactate, produce breast milk, the milk contains spider silk. The spider silk is pretty incredible. It's as strong as steel, but so light that a strand long enough to circle the Earth would weigh less than 500 grams. So there are thousands of uses for such a unique material. Scientists have used genetic modification to introduce genes that glow under UV light, such as from jellyfish into other animals for medical research. This may seem totally useless, but actually, being able to see certain cells in the dark has led to a lot of scientific discoveries. Glow-in-the-dark mice and fish have helped scientists make new breakthrough research into treating cancer, Parkinson's, and spinal cord injuries. And glow-in-the-dark cats have been used for HIV research. Crops can also be genetically modified to make them more pest resistant, to have nutritional benefit like additional vitamin A. To learn about GM crops, watch this video. So there we have genetically modified organisms that have had their DNA modified for some different uses.
So let me just get your view at this point. At this point, are you thinking that this is a very useful technique, or are you still cautious as to what it's about? What do you think?
For me, I think it has its benefits. However, I was doing some reading beforehand and watching all of this now. I found out that there are cases when they insert human genes into crops and other animals. And one of the concerns was how long before you consider something human after having so many human genes in it. And that seems like a very big question, and it seems like something important that needs to be dealt with and answered. So it has its place, but there's a limit.
Okay, well, that is a very good answer. That is probably the safest answer to give, to say that yes, it has its place. We cannot deny that there have been benefits, but we also need to be able to be cautious as to what are some of the harmful effects that we could experience from it. Let me just run through this one before I give you a short break. I hope you're seeing this one talking about some of the genetically modified organisms, persons that are pro-GMO, persons that are against it. But really, at your stage, you don't need to be for or against. You just need to be able to know points for both of them, okay? So genetic engineering is improving the quality, nutrition, and the yield of crop, plants, and livestock. Therefore, it is solving the demand for food in the world. For example, golden rice. They have been able to genetically modify the rice that produces large quantities of beta carotene in the endosperm. Human cells can then convert the beta carotene into vitamin A. So it's called golden rice. It has the same yield, it is pest resistant, and it has the same original eating qualities as the original varieties. But why are they engineering the rice so that it can have vitamin A? That's because in a lot of countries where there is vitamin A deficiency, it can lead to blindness. And vitamin A is fat soluble, found in fish, dairy, liver, etc. So if you're in a country where you're facing starvation, right? Like in Somalia, Yemen, one of the main things that they will get is rice. But rice is mostly made up of starch. So they might get rice, they'll barely get meat. So they won't get a lot of protein, and they're saying that a lot of these persons can suffer from vitamin A deficiency. So since rice is the main thing that they would get in these poverty-stricken areas, they're saying, why not enhance the rice so that it has vitamin A, so that when they eat it, they won't be deficient in this vitamin. So the rice that has been given to a lot of these countries with starvation problems has been modified so that it's more nutrient rich than just the regular rice, right? They said that they have also introduced the gene for carotene production into rice, daffodils, common soil and they have also inserted it into the common soil bacterium. Now, what are some of the ethical implications with this same golden rice here? Some organizations have condemned the golden rice. It is saying that that's the wrong way to solve poverty. Why don't you solve the political, the cultural, and the economic issues that help to lower poverty so that people can actually have a more varied diet? So that's one opinion, right? And this is showing you the golden rice versus the regular rice. So that has been one thing. Another, this is a question, so that's why I'm skipping over it. The other thing now, which they were able to alter as well, was the salmon. So there is this Atlantic salmon. So they injected growth hormone into the egg of the Atlantic salmon. Now, the growth hormone regulating gene was taken from the Pacific Chinook salmon, and they got the promoter from another species of fish called an ocean pout. But anyway, what they did is that salmon is normally a fish that will only grow in spring and in summer. But once they injected it with the growth hormone, the salmon was able to grow all year round. So they were able to reach market size in 18 months as opposed to 3 years if they were unmodified. So basically, these genetically modified salmon had genes inserted from 3 different species to create this genetically modified salmon that would be available in larger sizes and would be available all year round, essentially. And they are saying that this can help to curb the shortage of food, right? So that is somewhat a good thing, okay? So this is another question. So these are just 2 examples of genetically modified organisms or genetically modified crops, really, in agriculture that that you really should know. If you don't remember any other examples, you really should remember about the golden rice, and you really should remember about the salmon. All right, and let me just show you this one quickly as well, about the pros and the cons of genetic engineering in terms of food. So there have been persons who have been advocating for genetically modified foods because they believe it can solve world hunger. But let's look at the pros and the cons. So the pros, they said, is that you can have faster growth rates. So animals and plants can be genetically modified to promptly mature. For example, crops can be engineered to increase the crop yield. There are crops being engineered to survive unfavorable conditions, such as high heat or low light. This gives farmers the ability to expand beyond the traditional locations and use the empty lands for other purposes. Animals can also be genetically modified to improve production. Dairy cows can be engineered to produce more milk. Sheep can grow wool faster, and animals can be bred for their meat, and they can be engineered to grow bigger and faster. The con against this one is that there may be less nutritional value. So there are concerns regarding the nutritional value of the products that stem from genetic engineering, from the fact that some products can grow big and quick at the cost of decreasing their nutritional value. Let me give you an example of this. So the chickens, they say in the US, the chickens are bigger now, but they have a higher percentage of fat than what they had before, in the years before. So they are bigger in size, but they have less protein content. They have more fat content. So they're like, oh yeah, really, you're growing them faster, but you're not really increasing the nutritional value of the food. So that can be a con. Another thing they said is that you can use genetic engineering to make pest-resistant organisms an extended life. So this genetic modification is mainly executed in plants. The animal or plant genes are altered to make them I suppose they mean durable and resistant to pests and diseases. plants being cultivated can have built-in pest repellents, which help reduce the need for harmful chemicals and other pesticides that cause damage to the water supply. So they're saying if you can engineer the plant with a gene that they can produce their own pesticide and kill the pests, that means you won't have to spray the plants with pesticides or insecticides, and so it won't contaminate your water supply. What is the the con against this now? They're saying the con is that you can also create risky pathogens. You can create viruses and bacteria that can adapt to the environment. So if a bacteria cannot get through the natural repellent of the genetically altered plant, there is a possibility that these viruses and bacteria will grow stronger and more resistant and negatively affect the non-genetically engineered plants and animals. So they are saying that you have to look at it from two angles. So yes, you put in the insect, the insecticide resistance gene into the plant, but they're saying that viruses can overcome that. With time, they will learn how to bypass that, and then these will now become super viruses or super bacteria that can go on and infect the regular plants that were not genetically modified. And that is actually happening right now, because especially when it concerns corn, everybody has been producing the genetically modified corn, because right now if you go and just plant regular corn, your plants are going to be susceptible to these bacteria that have evolved and are very super bacteria that even if you use a regular pesticide or whatever, it doesn't kill them, because everybody else is using genetically modified corn. You have to fall in line because your crop yield is going to decrease, right? So that is another con. A pronoun. Oh yes.
Miss, if you put the pathogen, if you put the pesticide, pest resistant such organisms out there on the market and people start eating it, doesn't it cause infection to the body?
No,
Or something?
No, not that specific one. There are some which are going to come up now, which you're going to find that there are some persons who are allergic to genetically modified plants. Not necessarily the ones that you have inserted the insect resistance gene into, maybe some other gene, but persons that have had allergic reactions to plants that have been genetically modified, or even toxic reactions to them as well. But really and truly, the Food and Drug Administration is supposed to make sure that these plants are tested thoroughly before they are approved to be sold to the public. But there are still persons who would get a reaction to them. All right, so the other pro was that new foods can be made. So currently, we can create new types of plants and animals by combining genes from different species. Scientists are taking a potato or soybean and giving the crop more nutrients to increase its value. You know, like some persons who only, vegetarians, and they eat a lot of soy-based products. So they can become deficient in certain minerals and vitamins if they have a diet which is solely based on soy or potato. So they're saying you can enrich these plants so that there's a lower possibility of these deficiencies. So what is the negative side effect to this? The goal of genetic engineering is to solve an issue by transferring genes to the organism that will help combat the problem. Sometimes this can cause side effects. For example, you can modify a plant to need less water, but that would make it intolerant to direct sunlight. And then there's also unfavorable diversity. Genetically engineered species that reach the wild can have a negative impact on the domestic species. Since species that have been modified tend to be stronger, unmodified species would soon disappear because of the introduction of these modified species. And then this would result in decreased diversity. And based on all we have been speaking about so far, we know that decreased diversity is nothing good, because that can set up the entire species to become extinct should there be an environmental change that it cannot adapt to. So just bringing these concepts to you so that you can have a balanced opinion so far as it concerns genetically modified organisms, right? So I'm going to give you the break now 10-minute break. After that, we're going to talk about gene therapy, which is when we use genetic engineering to actually treat genetic diseases. And again, that also has pros and cons, right? So just take a 10-minute breather. Remember, do not sign out. Walk, drink some water, and come back and we'll continue. All right. I'm gonna start discussing gene therapy with you. So that's what I'm gonna show you next. So hope everybody's back. I'll soon show you the video. This is now talking about another use of genetic engineering, which is to deal with gene therapy. Now, this is possibly the more controversial part of the whole thing. And also, I have sent you an email with the folder that has all the notes for the genetic engineering so that you can download them and be able to read them through. Because as I said, today is really just to get you to understand the content of what it is about. And by next week, we can cover the questions based on this topic, right? So showing you our video now. So we've looked at the process of genetic engineering so far. We have looked at genetically modified organisms, genetically modified crops and animals. And now this is another branch of genetic engineering, which is gene therapy. So please watch so you can have an understanding of it for our discussion.
Hi guys, in this video we'll be looking at an introduction to gene therapy, using gene therapy to treat SCID, the method of gene therapy, the ethics of gene therapy, and then we'll finish with a summary. So this video is all about gene therapy. So some of the genetic diseases that happen in humans occur because of the presence of a faulty or what we call mutated version of a gene or allele. So remember that every protein in the body is coded for by a gene, and that gene has to have the correct sequence of DNA bases to code for the correct protein. Sometimes we can have mistakes in that sequence caused by a mutation. So mutation, remember, is the change to that sequence of bases, which usually leads to a faulty version. So we get a faulty allele and therefore a faulty protein, and that can cause genetic diseases. So that means that many scientists are looking at different ways to replace that faulty allele with a healthy version of the allele, and this process is known as gene therapy. So a therapy is when we're trying to fix something or help or cure a disease. Gene therapy simply refers to the idea that we're changing a faulty gene and replacing it with a healthy gene. So we're actually changing the DNA code of the cells of that individual. So for a definition, gene therapy is a mechanism by which genetic diseases are treated or cured by masking the effect of a faulty allele by the insertion of a functional allele. So we're taking the faulty allele, we're replacing it with a healthy allele, which can make the healthy working protein. One example of using gene therapy has been used to treat a disease called SCID. And we've been using viruses for gene therapy to treat this genetic disorder. It's SCID, which stands for Severe Combined Immunodeficiency. This disease essentially means that an individual has a very poor immune system. That's what immunodeficiency means. And it's a very severe condition. And early on, people who had this disease had to live in a sterile bubble, if you like. Because if they come into contact with any pathogens in the outside world, their immune system is not able to fight them off, and they would die very, very early. So they have to be kept in this bubble, which has air without any pathogens in it. So it's obviously a very poor quality of life. There are different forms of this disease, but one form of SCID can arise due to a defect in a particular gene. And this gene codes for a particular protein, which is an enzyme, and that enzyme is called adenosine deaminase, or ADA. So don't worry too much about what this enzyme does for the moment. Just be aware that the gene we're talking about here codes for ADA, and the mutation which hits ADA leads to this condition. So the problem with this mutation is that T cells, which are involved in the immune system, aren't able to proliferate, and this weakens the immune system. So a really important feature of the immune system is where we have a T cell or a T lymphocyte recognizing a particular pathogen. And to get rid of that pathogen, normally the T cell would divide or proliferate into many different T cells, which can then tackle that pathogen as one big army of cells. In this mutated version of ADA, which doesn't work anymore, this process can't happen, which means the immune system can't fight off pathogens. So the current treatment is with a bone marrow transplant or an injection of ADA. So in injections of ADA, essentially what we're doing is inserting the ADA protein as a replacement therapy, just in the same way that we would inject insulin for diabetes. Bone marrow transplants involve taking bone and taking the soft tissue within bone and replacing it with healthy bone marrow from a donor. And the bone marrow is involved in making T cells, as well as other cells in the blood system, like the red blood cells, so then it's able to make more healthy immune cells. But now scientists are starting to use gene therapy to treat SCID, and by the definition of gene therapy, this means they're inserting a healthy version of that allele that codes for ADA straight into the T cells. So remember, people with SCID have a mutated version of the ADA allele. So what we do is we take the healthy version, knowing what the healthy sequence is, which does work, inserting it into the nucleus of the T cell, and then of course the T cell can make ADA. So we're replacing the genetic code, and this is therefore gene therapy. So let's go through the method of gene therapy in treating this disease. The first step in using gene therapy to treat SCID is isolating the healthy ADA allele from a healthy cell tissue. So remember, we're trying to replace a faulty allele with one that actually is the correct sequence. We're not going to find this individual who has the disease because every one of their cells has a mutated version of that gene. So we take healthy tissue, so someone who does have the actual correct allele, and we simply take the allele out. So we go into the nucleus, we go through the DNA, and we find the ADA gene, and we find that it's a healthy allele. What we need to do then is get this into the individual who has the disease. So the first thing we do is the ADA allele is inserted into retroviruses, and retroviruses replicate by copying their DNA into the host cells. So just to explain what that means, a retrovirus is a type of virus that will go into a host cell, and it will enter, and eventually it will enter the nucleus. And what it will do is it will put its genetic material into our own genetic material so that our own cells can make more retroviruses. They replicate, they burst out of the cell and then spread around the body. So any DNA that's found in the retrovirus will be put into the nucleus of the host cell. So the ADA allele that we found, the healthy one, will be put into this so that it will be put into our own DNA. So in order to mix them together, the patient's T cells are extracted from their blood, and the T cells are mixed with the retroviruses. So remember, these T cells have a faulty version of ADA in their nucleus, but the retroviruses have a healthy version of ADA carried in their structure. So these are mixed in with the T cells in specific equipment. And of course, the viruses want to infect the T cells, so the retroviruses inject a copy of their healthy ADA allele into the T cell's nucleus. So the viruses begin to approach the T cell, because many retroviruses usually infect T cells anyway, and then of course they inject their material and it goes towards the nucleus. Nothing always works all of the time, so a small proportion of the T cells will successfully incorporate the ADA allele into their genome, and some of them just won't happen to. So that healthy ADA allele is now incorporated into our own DNA. And remember, all of our DNA is found in every nucleus of every cell in the body. So that means that these T cells that have taken up that allele are able to produce the functional ADA enzyme. They don't have a faulty version anymore, it's been replaced with a healthy one. So when they transcribe this and translate this, they make the protein ADA, which means that our immune system can then work. So now what happens is those T cells are reintroduced back into the patient's blood, and they're able to proliferate. So essentially, the T cells that did take up the allele are successfully treated. We put them back in the blood, and T cells will proliferate in their life anyway. So they become a massive population, which means all the T cells in the blood will be able to make ADA. However, T cells don't live forever. The T cells do not live more than about 6 to 12 months. They will die after that. So because they'll die, the T cells that the individual is making will still have the faulty allele because they're originally coming from the patient's cells. So this procedure has to be repeated at regular intervals. That pool won't last forever. There are some ethical concerns around gene therapy. It has many ethical issues that we have to consider. There are concerns about the potential impact that inserting the gene might have on the functioning of other genes. When we think about genes, we often think of them in isolation, but that's not actually the way to think about them. We often just think that one gene codes for one protein, but actually many genes code for proteins that interact with other genes. It's a very complicated interaction, and many genes control the expression of other genes. And we don't know fully if the introduction of a healthy allele might interact with other proteins. And if they're healthy proteins, they may be downregulated. So we have to be aware of all the different interactions that might happen when we carry out gene therapy. There's also a big concern that the replacement of non-functional alleles with healthy alleles could be a slippery slope towards designer babies. So obviously it's important to make sure that natural selection still happens and that babies are born naturally. If we start mixing around different alleles and altering them, then this may impact the population in the future. So designer babies could be made by replacing less desirable alleles for more desirable alleles in early stage embryos, even if it's not crucial to their survival. For example, if parents were specifically asking for a baby with blue eyes, they might introduce the blue eyes alleles and delete the brown eye alleles. Of course, if we mess around too much, this could have consequences in the future. Hey guys, I hope you enjoyed the video. If you're looking for an amazing A-level...
Alrighty. Let us start our discussion for this one. So gene therapy is on another level now. Let's just go back through it. So let me hear from you. What did you grasp from it? What is the purpose of gene therapy? What is the purpose of gene therapy? What are they trying to achieve? Anybody? Let me unmute you guys. Anybody can talk. What is the entire objective of this therapy?
To prevent certain diseases like SCID.
Like SCID? Well, certain diseases that's still a little bit too vague. What type of diseases specifically does it try to help persons with?
SCID.
SCID. All right, that was just one example of a specific disease that they had been discussing. Oh yes, but the category, yes, that's what I wanted to hear. So the category of diseases that it is designed to help persons with are genetic diseases. Diseases which were due to diseases which you have inherited and which are due to faulty alleles, right? So SCID was the one he was discussing in the video, but SCID is not something you see in Jamaica. Can you tell me of an example of a, that is more relevant to us in the Caribbean, of a genetic disease that we could possibly use this genetic engineering to cure?
Is vitiligo one?
Vitiligo. That's not no, that's not a genetic disease. That's an autoimmune disease.
Okay.
And so would be like sickle cell?
Sickle cell, yes, sickle cell. That is our classic genetic disease, which is due to one faulty allele that would be a candidate for this type of therapy. So if it is that the disease is caused by multiple genes, then it is going to be very challenging, because it is challenging already to solve the problem when there's only one faulty gene, much less when you have several. So we wouldn't try to use gene therapy to solve like diabetes or something like that, because diabetes is as a result of multiple genes being faulty, right? So it would be better for inherited or genetic diseases that are only due to one faulty allele, of which sickle cell anemia is a classic example. Also, hemophilia would also be a classic example of one that would be a good candidate for this. In the example that he described, he spoke about SCID, which is severe combined immunodeficiency, and there's also another condition that you might come across called cystic fibrosis, which is also due to one faulty allele. So the whole purpose is that we are trying to help persons who have inherited a disease due to a faulty mutated allele. So that is the whole aim of the whole process. Now, the whole objective of doing this is that we need to be able to replace the faulty allele with a good copy of the gene. So that's what they are trying to do with gene therapy. So you try to find where the allele was faulty and see if you could replace it with one that is good. Okay. Now, in theory, it sounds nice, it sounds simple, but to actually put that into practice, it's very difficult. So it says that gene therapy is a mechanism by which genetic diseases are treated or cured by masking the effect of a faulty allele by the insertion of a functional allele. And then they were describing this classic condition, which we would want to cure, definitely, which is a severe combined immunodeficiency. I don't know if you have ever heard of this movie about this called Waterboy or something, but it was a movie based upon the same condition where and I think there was one recently out where this young lady, she had to stay inside all the time.
Everything.
Yes, everything, yeah. And her mom, you wouldn't let her out because I think she would have suffered from the same condition here. So you have to like keep them away from all germs because they can't mount an immune response against it. So definitely this is a disease you'd want to cure people of. It's deadly and they don't live very long, right? So if they eventually kind of did some studies and they found that some persons that had the SCID, it was really because they had a defect in one particular gene that codes for a specific enzyme. So they were saying that, okay, if we can fix this faulty gene, then the person can produce the right enzyme and they won't have this problem. So they found out that the faulty gene was actually present in T cells, and T cells are lymphocytes that help to boost, well, they're the main lymphocytes that help us to defend ourselves against diseases. So because the lymphocytes had the faulty gene inside of them, they weren't able to mount an effective immune response. So they were trying to see if they could somehow deliver the healthy allele for the enzyme to all the T cells, and so that the faulty one doesn't get expressed. So they tried giving people bone marrow transplants because you know the bone marrow is where the T cells are made. So they tried injecting the enzyme into the bone marrow. Then it worked to some extent, didn't work to some extent. So then they said, all right, why don't we try injecting the gene for the enzyme in the actual T cells themselves? So that's what they ended up trying, they started to do with the gene therapy. So then they had to figure out how they were going to get the gene into the cell of interest. So first they had to isolate the gene from the healthy tissue, and then they needed to get the gene into the cell. So they figured out that the best way or the best vector that they could use to get the gene into the cell would be to use a virus, a retrovirus. So a retrovirus is a virus that can infect cells, and they are able to inject their DNA into your DNA and force you to actually start making their proteins. So retroviruses would include like HIV. So because they knew that these viruses were already very good at injecting their DNA into host cells, they said, okay, if we could put this gene into the retrovirus, then we could get the retrovirus injected into our cells that we want them to inject them into. So that was the principle of it. So they got the patient's T cells and they mixed them with the retroviruses that contained the corrected gene. And of course, the retrovirus would infect the T cells and would inject their DNA inside there. So a small proportion of the T cells they were actually able to get the corrected gene, the ADA gene, into the T cells so that they could actually make the protein. And they were able to produce a functional enzyme, and it did help. And then they put it back into the patient's blood so that they would multiply and that would help the person. But what were the challenges that they found with this down the road though? He said it they did say it at the end, that the T cells that they fixed in the lab, they don't live for more than 12 months. So the ones that are going to be produced after 12 months would not have been fixed. And so the person would end up back at square one. So they're like, you'd have to do this treatment every 12 months. And then they were like, okay, there were a lot of ethical considerations with the gene therapy, the fact that we cannot alter genes. And they are saying that what if you you are trying to insert a gene to fix a problem, but what if you insert a gene and that creates a problem? Because maybe the gene that you have inserted might have gone in and disrupted other genes from working. So that was a concern they had with the gene therapy so far. And then they're like, even though it might seem like it's a very useful technique now to try to help these persons, it can also be manipulated. And you can also use it to try to have designer babies, where you decide what traits you want your baby to have and for them to genetically engineer the child to have those traits. By replacing less desirable alleles with more desirable alleles at the embryo level, right? So they're saying that this technology could be manipulated for that reason. So that is a possible downfall. But there are some other downfalls with gene therapy, which I will go through now. A second. So the genetics, gene therapy. So bring those up for you. Okay. So this is a document that is just describing the whole process of gene therapy again. Because as I said, I'm sending you the different documents so that you can have the information, right? And then we will look at some of the downsides. So gene therapy is a treatment of a genetic disorder by altering a person's genotype, which is that you are inserting functional alleles of genes into cells. In order to do this, you need a vector. So vectors include like viruses, retroviruses, lentiviruses, HIV viruses. These are all retroviruses. They are designed to infect cells and inject their DNA into cells. Other ways you could introduce it is by using liposomes, which are small spheres of phospholipids, or you could just inject the naked DNA into the cell. Retroviruses insert their genes randomly into the host genome. If the gene is inserted into another gene, or if it is inserted into a regulatory sequence of a gene, it could activate a nearby gene and cause cancer. So this is one of the challenges that they discovered when they were actually doing gene therapy. So in the early parts of them doing this, they realized that some of the persons that they were trying to help actually developed leukemia and various other forms of cancer and actually died from the cancer and wasn't cured from the disease that they were trying to treat, because they're saying that yes, you're trying to insert a gene, but the virus is not very specific as to where it is going to insert the gene. The virus is just going to insert it anywhere, and they're saying that it might insert it in the wrong place and end up leading to a cancer. So that was one of the problems with the gene therapy down the road. So this is an example of the retrovirus. So lentivirus inserts its genes randomly into the host genome, but this virus can be modified to inactivate replication, for example, HIV. And you have another set of viruses that do not insert their genes into the host genome, and gene is not passed to the daughter cells when the cells divide, and the virus can be used successfully with long-lived cells. And then it talks about the SCID again that we saw, which is a crippled immune system. Sufferers may die at infancy due to normal infections, and some of them have an inability to make adenosine deaminase. So T lymphocytes of sufferers are removed, and normal alleles of the ADA gene are introduced into them using a virus vector. This is not a permanent cure. Another disease that they tried it with was cystic fibrosis, which is a genetic disorder in which you produce abnormally thick mucus in the lungs and other body parts, and it's caused by a recessive mutated allele of the gene for a functional transporter protein or chloride ions. and it's due to a deletion. So anyway, the point is that the person doesn't produce watery mucus, and so they get plugs all over the body, in their lungs, everywhere. And they believe that if you can use gene therapy, then you could replace the faulty gene, all right? So this is for somebody living in the bubble, person with SCID, as we saw before. So, and this is them showing you about what happens with cystic fibrosis when you have thick, sticky mucus blocking up everywhere. So these persons have mucus accumulating in the lungs, which makes it difficult for them to breathe. They also have their pancreatic ducts being blocked up, and so they can't secrete their enzymes, and it blocks up even the sperm ducts, so they are infertile. So the gene therapy is designed for these persons. You can either have somatic gene therapy or germ cell therapy. Somatic therapy is when you're targeting body cells, and germ therapy is when you're targeting reproductive cells. So some persons have been able to insert the correct gene into the cells by doing electrophoresis or electroporing to get it inside of the cells to try to correct that problem. And this is just some questions coming down below. But that is the basics of gene therapy. We kind of have seen some of the pros and cons already, but I'm going to look even some more at some more pros and cons in a minute. The advantages and the disadvantages of gene therapy, so that if you're asked about genetic engineering or if you're asked about gene therapy, you are able to give a balanced opinion. So this document now that I'm showing you is talking about, first it has an introduction, and then it gives a list of the pros of gene therapy, and then it gives a list of the cons. So this is the last part of what we're going to go through. Also, guys, I still have your questions that most of you submitted to Mark on natural selection and speciation. I'll be marking them and sending them back to you in the week. And then I may send you, if I find an MCQ on genetic engineering and gene therapy, I'm going to search for it. I may send you one or two MCQs. And what you need to do is that you need to download a PDF reader or a PDF, anything that can allow you to annotate the PDF, anything that allows you to shade or write on the PDF, so that you can select the answers and send them back to me. Most of you would have seen some announcement that CXC is planning to give a multiple choice exam. So for that reason, we can't depend on just the same ones that we see in the past paper book being recirculated year after year, because I think more or less they're going to have to write new questions and add to the pool. So I'm also trying to make you exposed to more questions, all right? So I'll be sending back the natural selection ones after I've completed the marking, and then look out for an MCQ or two on gene therapy and genetic engineering. All right, let's go through the list of the pros. So the first pro so far is that it provides hope for those that may not have had any in the past. So about 3% of American children are born with a genetic condition which requires gene therapy as a way to treat the issue. At this time, the diseases and disorders which are present in this population will take the life of the child before there is an opportunity to correct the condition. Birth defects are the leading cause of newborn deaths in the United States, with as many as 1 in 5 children suffering from them. So advances in gene therapy could help to correct these issues instead of forcing parents into a heartbreaking scenario. Gene therapy could also change the perspective that people have about diseases. Roughly 10% of all persons are Americans, this was an American article, are affected by rare diseases or condition in any given day. Approximately 33 million people are suffering from disability that is attributed to their genetic profile. So the promise of gene therapy is that it can reduce or eliminate the pain and discomfort of these abnormalities. And 80% of the diseases that we know impact human health in negative ways have a genetic foundation. So if we can replace the cells or chromosomes that are at fault, then it becomes possible to offer relief. Gene therapy could offer potential new discoveries. So our world is a better place when there is an emphasis on diversity. So when we have effective gene therapy treatments that can save lives or prolong them, we're adding strength to our existence. There will be more opportunities for research. New ideas could lead to critical discoveries and relationships that can lead to future generations that experience these benefits as well. Going on to the next one, gene therapy could be used in different ways to improve life. Right now, the focus of gene therapy research is to provide solutions for people who are suffering from specific illnesses or disease. But when we begin to experience successes in this field, then the information we learn can be applied to other treatments as well. So gene therapy could be useful in the treatment of infertility issues, for example. It says gene therapy doesn't only apply to human options. It is essential to remember that the benefits we can experience as humans can be applied to other forms of life as well. So the technologies we create from this research could help us to grow crops that adapt more effectively to changing climate conditions, etc., etc. And then this is just a repeat of before, that it helps to improve the quality of life for some people. It helps to treat the untreatable diseases, etc., etc., right? So 7 pros of why gene therapy is good. But now let's look at the cons of it now. So gene therapy does not have a reliable delivery method. We have to depend on retroviruses as our delivery systems as the most common way for gene therapies to be delivered to patients. And the problem with this option is that the enzyme used to encourage the transfer of genetic data can be eliminated by the immune system before it has a chance to work. There could be issues with cell division or replication that limit the effectiveness of the treatment. And when there is a noticeable change to the cell, the body might attack itself without the presence of an immunosuppressant. So until we can remove and replace genetic data with more reliability, the success stories for gene therapies will be a hit or miss. Next con, gene therapy is an expensive procedure. There are several gene therapy options which are available right now, but they come at a steep price. If you use Luxturna to treat both eyes as a way to treat blindness, the final cost could be more than 1 million US. Even the affordable options in this field starts at 200,000 US per treatment. That's why many patients wait for clinical trials to begin and then apply for a spot in one of them to receive the help they need. Most health insurance plans will not cover the cost of these procedures because of their uncertainty. Gene therapies also require ongoing treatments to be effective. Many people have found that the benefits of their gene therapy treatments begin to wear off as soon as they were no longer able to take their medicine or visit their doctor for treatments. It can be a lifetime course that someone must follow to reduce or eliminate the genetic issues that hold back their health. Unless you can keep taking the products, which are often priced above $100,000 per treatment, then you will experience a reversal in your condition. And next con, gene therapy may not be able to adapt to a changing world. It has taken less than a century for prescription-grade antibiotics to no longer be as effective for the treatment of bacterial infections as it once was. Antibiotic resistance can impact anyone at any age and in any country. Sometimes it also occurs naturally. But the most common reason for this issue is that antibiotic misuse has led to a growing number of infections, including pneumonia, tuberculosis, and salmonellosis being challenging to treat because the medicines are no longer effective. This issue could also occur with gene therapies too. We have already seen people begin to have their progress reverse itself when they stop following their treatment plan. So over the next couple of generations, the body could start resisting this option too. Another con they say is that gene therapy might just be delaying the inevitable. Let me just go to this part. This is like a story. But said the National Institute of Health approved the first human gene transfer study in 1989. Through 2006, there were 800 gene therapy studies that involved 5,000 patients. And in those 17 years, the total number of approved therapies was 0. The only success story was a cure for a bubble boy disease that also caused leukemia thanks to the virus that delivered the treatment. So they're saying that this thing is not even so effective because they treated 5,000 patients and only one patient was cured, and then the one patient that was cured ended up developing leukemia at the end of the day. So it's not very successful. And the other two cons, less so about talking about change the way towards polarization, how we think about competition and all of that. But this whole thing, as I said, is just to get these thoughts into your head for you to, at this point, be able to have a balanced discussion. You, at this point, should know exactly what genetic engineering is all about. You should know how it is done. What are the benefits of producing genetically modified plants and animals? And then we just explored this other aspect of it called gene therapy. When would it be useful? How is it done? And what are some of the possible harmful effects of it? And that in all is basically what it is required of you to learn about your genetic engineering. There are only one or two things that I haven't discussed, but I have placed in the folder for you to download, that you can take a read for. Take a read of them all so that you can have a nice balanced opinion. But that's basically it. So we have covered the content, and all I need for you to do, for persons who hadn't sent me the questions from last week, I hope you send them to me by tomorrow so that they can be marked. And your job is to read over this information that we would have gone through today. I have emailed you a folder that has all the notes in it. So your job is to read it over and be prepared for next class when we will be doing questions on genetic engineering. Do you have any questions on what we've done today before we go? Is it clear for the most part? Guys, is it clear? Do you have any questions before we go?
Lisa, how are you going to email us the documents, or
Yeah,
are you
man,
going to
I did. I sent it. I sent you the emails already.
Miss, the website that you're using, are you going to send us the link or is it in the email?
No, all of what I used were documents. What you meant, the videos? What do you mean by the website?
Miss, I saw something on A level, something like that.
No, it was a PDF.
Oh, okay. And you sent that?
Yeah, I put all the documents I used today in a folder and I sent the email to you for you to access the folder.
Okay.
Mm-hmm. So that's it for today. And I'll see you, some of you shortly for Chem. Alrighty? So enjoy the rest of your day. And remember to read over the documents, all right?
Okay, Miss, bye.